Hello everyone and welcome back to my channel. Today's video is about glycolysis or EMP pathway. So this is the glycolysis pathway and we know glycolysis has total 10 steps. So in today's video we will learn all these 10 steps in detail. So let's start. First is glucose. So glucose is the substrate through which glycolysis cycle starts. So glucose is a 6 carbon compound. In the first step, it gets converted to glucose 6 phosphate, which is also a 6 carbon compound. Now here, one additional phosphate has been attached to the 6th carbon of glucose. So where did this one phosphate come from? So here ATP is consumed or utilized and converted to ADP. That means one inorganic phosphate is free. So it gets attached to the 6th carbon of glucose and it becomes glucose 6 phosphate. And the enzyme present here is hexokinase. This is the first step. Second step is glucose 6 phosphate getting converted to fructose 6 phosphate. Now there is no change. Only glucose has been converted to fructose. And we know glucose and fructose are isomers. So here the enzyme is isomerase. This is the second step. Third is fructose 6-phosphate getting converted to fructose 1,6 bisphosphate. Now here one additional phosphate is attached to the first carbon of fructose. This again phosphate came from utilization of ATP which converted to ADP and one inorganic phosphate written as Pi gets attached to first carbon of fructose. Here the enzyme is phosphofructokinase. This is the third step. In the fourth step, fructose 1,6 bisphosphate gets converted to glyceraldehyde 3-phosphate. Now this is a 3 carbon compound. Now all compounds after this are 3 carbon compounds. Here the enzyme present is aldolase. This is the fourth step. Now glyceraldehyde 3-phosphate gets converted to dihydroxyacetone phosphate, which is also a 3 carbon compound. Now there is a common thing. They have another name called triose phosphate and this also has triose phosphate. This shows that these both are isomers. So here again the enzyme is isomerase. Now out of these two compounds only one will continue the glycolysis cycle. So in this chart you can see glyceraldehyde 3-phosphate is continuing the process. Now this is the fifth step. In the sixth step, glyceraldehyde 3-phosphate gets converted to triose bisphosphate, also called as 1,3 bisphosphoglyceric acid. Here NAD plus gets converted to NADH plus H plus. This is also written as NADH2. And the enzyme present here is dehydrogenase because this reaction is a dehydrogenation reaction. This is the sixth step. In the seventh step, 1,3-bis-phosphoglyceric acid, which is a 3 carbon compound, gets converted to 3-phosphoglyceric acid. Here, one phosphate which was attached to the first carbon is removed. That means it was used up somewhere. So it was used to convert ADP into ATP. So ADP plus one inorganic phosphate gives us ATP. So here ATP is produced. Here the enzyme is phosphoglycerate kinase. And this is an important step. It is called as substrate level phosphorylation. So this is the seventh step. In the eighth step, 3-phosphoglyceric acid gets converted to 2-phosphoglycerate. Here the enzyme is mutase because the phosphate which was present at the third carbon got converted or get replaced to the second carbon. So here mutation has taken place. This is the 8th step. In the 9th step, 2-phosphoglycerate gets converted to phosphoenol pyruvate. And here the enzyme is enolase. And water gets removed from this molecule. This is the 9th step. And the last 10th step is phosphoenol pyruvate, also called as PEP, gets converted to pyruvic acid, the final key product of glycolysis. Here also ADP gets converted to ATP, that is ATP is produced. This is the 10th step and the enzyme here is pyruvic acid kinase. Now from the 6th step you can see these compounds, before that this 2 into is written. This means all the compounds are present in 2 molecules. So 1,3-bis phosphoglyceric acid, it's 2 compounds. 2 compounds of 3-phosphoglyceric acid, 2 compounds of phosphoglycerate, 2 compounds of phosphoenol pyruvate. So at the end also 2 molecules of pyruvic acid are formed in the end. So this was glycolysis or EMP pathway. So we have seen 10 enzymes. First was hexokinase, second isomerase, third phosphofructokinase, fourth aldolase, fifth isomerase, sixth phosphoglycerate, seventh mutase, eighth enolase, and the 10th pyruvic acid kinase. So these are the enzymes. And these were the 10 steps of glycolysis. Now here are some important things to remember in glycolysis on which questions can be asked. So in the glycolysis, 2 ATPs are consumed, that is utilized, and 4 ATPs are produced. Therefore, the net gain of ATPs will be 2 ATPs. Out of the 4 ATPs formed, 2 are utilized in the steps. So 2 ATPs is the net gain. The number of NADH2 produced are 2. And we know one NADH2 gives 3 ATPs. So total 6 ATPs from 2 NADH2s. So in the last, the total net gain in glycolysis is of 8 ATPs. Now, in the glycolysis, some steps are irreversible and some are reversible. So here, the irreversible steps are first, third, and the 10th step. Reversible are all other rest steps, that is second, fourth, fifth, sixth, seventh, eighth, and ninth step. All other are reversible reactions. Now, at which steps ATP was consumed and at which steps ATP was synthesized. So ATP was consumed at first and the third step, and ATP was synthesized at 7th and the 10th step. So these are some important points to remember on which questions can be asked. So if you like this video, click on the like button and don't forget to subscribe my channel to get notifications of more such new videos. Thank you.